Welcome back, BIO178. In the last video, we talked about ways you can identify sex bias dispersal by comparing populations. And here in this video, we're going to talk about ways you can use individual level analyses to determine whether sex bias dispersal is happening or not. In other words, you can look at an individual compared to the population as a whole, and in that way, determine whether sex bias dispersal is occurring. So one of the ways you can do that, we've briefly touched on a little bit before. You can use assignment tests, and what you can do is compare the numbers of males and females that are assigned to a population different than the one they were sampled in. If that's the case, that means they were a migrant that came from somewhere else. So here, what I'm showing on the right, this is a structure plot, which we've talked about before in several previous modules. And this is based on a fish. What fish was it? Sturgeon, Atlantic sturgeon. And what we can see here in the top structure plot, remember each vertical bar is a different individual, and we have different sampling locations here on the x-axis of the structure plot, the bottom of the structure plot. So what I mean by assignment tests, so all of these individuals in this red block here, they were sampled in this Canada region, which is right here. However, if we look at this individual, for instance, the one that has the high yellow bar, that is actually genetically most similar to this other population that's right here. So we would assign that individual to a different population than the one it was sampled in. That would indicate that particular individual was a migrant. And we can do that for males and females and determine whether the number of those migrants is different for males and females. If more of the migrants were males, for instance, we would infer male bias dispersal. So how we do this, first of all, each individual is assigned an assignment index. That's abbreviated as AIC. That lowercase c indicates that it's a corrected assignment index. We're not going to go into the details, so don't worry about it. But if you see that term AIC, that's what that means. It's an assignment index. And that basically is telling you the probability that the individual you sampled is from its population where you sampled it, or if it's from a different origin population. And when you're interpreting this, specifically a negative value when you're calculating these AICs indicates it's a rare genotype relative to the population it was sampled in. So that yellow individual we talked about here, that individual would have a negative AIC value because it has a rare genotype relative to the population it was sampled in, the rest of that red Canada population. And therefore, that individual is likely an immigrant. And so here's an example of that. They use these assignment indices in the white-toothed shrew, which is found in Europe. And when they plotted those AIC values, which are shown here on the right, so we have the number of individuals here on the y-axis, the AIC value here on the x-axis, and then we have females in the black color, males in the lighter gray color. Remember, a negative AIC value indicates that individual is an immigrant from somewhere else. And so if we look here, so if we draw a marker here for this is where the 0 AIC value is, everything this way is negative, we see that overall there are more females that are assigned to negative AIC values compared to males. So in this case, what we would conclude is that females are the ones that are moving between populations. That indicates a pattern of female biased dispersal. Another method we can use is called spatial autocorrelation analysis. So what we do here is essentially calculate a correlation coefficient, abbreviated by lowercase r, and we calculate that between genetic and geographic distance. This is very similar to when we talked about calculating or determining whether isolation by distance is happening in one of the previous modules when we talked about doing a Mantel test. This is very similar. So we calculate this correlation coefficient between genetic and geographic distance. And then what we do in these type of spatial autocorrelation analyses is we plot that correlation coefficient on the y-axis and we plot it against, on the x-axis, the distance of these different populations in relation to each other. And what we do then is, in this particular situation where we're trying to determine if sex-biased dispersal is happening, we conduct one of those analyses separately for each sex. So we would do one spatial correlation analysis for males, create a plot, do one for females, create a plot, and then we compare them. So, and the pattern in those plots will look different depending on whether that sex is dispersing or not. So the more philopatric sex, that's the one that stays close to home, will show positive spatial autocorrelations in the first distance class. So in this graph here on the right, the first distance classes, those are the shorter ones. And what we can see is that the correlation coefficient is higher in those first distance classes here. The value of R is higher. That indicates that the sex that was used to create this particular plot is staying closer to home. In contrast, the sex that's dispersing and moving between populations is going to show no spatial autocorrelation pattern. And that will look something like this. Here on the right, we still have distance on the x-axis. That correlation coefficient is still on the y-axis. But now, instead of seeing a pattern like we did before, it's just all over the place. There's no discernible pattern. That indicates that whatever sex was used to create this plot is the one moving between populations. So now we get to talk about wombats, which, as I said, apparently I get very excited about because one of my students from 3 years ago still sends me wombat things when she sees them because it makes her think of me. And I do like wombats. They're pretty cool. They have cube poop, if you didn't know that. If you aren't aware, Google it. They're one of the only animals that have poop that's shaped like a cube. Okay, so what they did in southern hairy-nosed wombats, to be specific, is they did these spatial autocorrelation analyses, and they did them separately for males and females, right? So what they found for males, which is the graph that's shown here, males are positively correlated in the short distance classes. So again, the y-axis here is that R correlation, the x-axis here is distance. And so what you can see is that in the short distance classes, here we see that same pattern where we see higher R values in the short distance classes. What that means is that males that are close to each other in close geographic proximity are genetically similar to each other. That means they're the ones that are staying close to home, they're born, they stay in that same area, and their relatives are surrounding them. The males aren't moving. In contrast, when we look at the females, which are shown here in this new plot now, there's no spatial autocorrelation pattern for the female. So again, if we look at the pattern here, distance is still the x-axis, that correlation coefficient is the y-axis. Now we see a pattern that's all over the place. What that indicates is that females are the ones that are moving between different populations. So here we would conclude that female biased dispersal is occurring in the southern hairy-nosed wombats. So one other way we can use individual type analyses to determine whether sex-biased dispersal is happening, this is one of the most precise methods. So this is using parentage analysis, which we've talked a little bit about already in this particular module. And parentage analysis is precise because instead of inferring whether males or females are moving by doing things like calculating relatedness or assignment indices, you can actually determine who the parents of a particular... Sorry, I don't know where that cut off. I record these on my iPad and a phone call came and interrupted it. So if we're doing parentage analyses, what we can do is determine the actual parent of a particular offspring, and based on the location of those parents and the offspring, we can determine whether that individual dispersed or not from where it was born. So that's why it's really precise. It's challenging though, because what you have to do is sample from a bunch of different possible source and sink populations. So in this example here, the example that they talk about in your textbook is, say we have an aquatic larvae. So this is a sea urchin larvae that's shown here. Say we sampled this larvae in population A. And we did genetic analyses, like we've talked about earlier in this module, and we determined that its parent, this is an adult purple sea urchin, these are found off the coast of California. If you've been in some tide pools, I've seen a lot in Southern California. I don't know if I've seen so many in Northern California, but there are sea urchins in California in tide pools. So if we determine that the adult parent of this particular sea urchin was found in population B, a different population, we would then conclude that that larvae was born in population B and transferred, moved to this new population, and that then would help us determine if there's sex-biased dispersal happening, because we also know the sex of the larvae. Again, it's challenging because you have to know which possible populations to test to see where the larvae came from and where it could possibly end up. So it's challenging, but they have actually done this successfully in black bears, for instance, in a recent study here. And what they did was, I think they sampled something like 2,500 black bears. So it was a really big study. It required a lot of sampling effort. What they found is they compared the distance between male offspring and their mother and between female offspring and their mothers. And what they found was that overall the geographic distance between a mother and a male bear was farther, indicating that males disperse farther, which would help us conclude that there is male bias dispersal happening. So in this graph shown here, what we're looking at is the proportion of dispersed bears. The males are in the black bars here, and the females are in the lighter gray bars. And we can see overall the proportion of males that dispersed is much higher across these different age classes, indicating that male bias dispersal is happening. So we've talked about a bunch of different ways you can determine if sex bias dispersal is happening using molecular data. One of the ways we can be really confident in our results is if we have what are called concordant results. In other words, if we do several of these different types of analyses, say we did assignment indices and spatial autocorrelation analyses, and both of those types of analyses give us the same information, say they both tell us that female bias dispersal is happening, we can then be more confident in our results because we have several different methods giving us the same answer. So an example of this was in bush hyrax in the Serengeti National Park, and they did 3 different methods in this study. They calculated FST values, they calculated they did assignment tests, calculated assignment indices, and they also calculated relatedness coefficients. And all 3 of these different tests indicated that female bias dispersal was happening, indicated that females were the ones moving between different populations. So I'll give you some time to kind of think about all of the things we've talked about so far. Think about for each of these 3 tests, what would the data look like if females are the ones that are dispersing and males aren't? So I'll give you some time to think about that, jot down some notes. You can pause the video if you want. I'll give you some time before I give the answers. All right, if you're still working, go ahead and pause the video. So let's start with FST values. So if we know that females are the ones that are moving between populations, that means that females are going to have low FST values. Remember, the lower an FST value is, the more genetically similar these different populations are. So if females are moving, the female-female FST values are going to be low because they're moving between populations. For the assignment tests, let's use a different color for that. For the assignment tests, if females are the ones that are moving between populations, females are going to have negative AIC values. Those indicate they're migrating between populations. And then lastly, for relatedness coefficients, we will find for the females, if they are moving between populations, they're going to have low relatedness. And that's because they're basically, females are being born, they're moving to a different location, and so they're not going to be related to the other females where they are sampled because they came from somewhere else. So it's an example of how you can use several different types of analyses to come to the same conclusion. That means it's more likely that the conclusion you came to is actually correct.